Breaking the wall of chemotherapy consequences. Qualified in a McMaster lab. Erin Westman, McMaster University, Canada.
We've heard a lot about cancer today, which is very important because worldwide, about 1 in 4 of us is expected to experience cancer within our lifetimes. In order to treat cancer, an important option that we have in our arsenal of tools is chemotherapy drugs. But as you've heard earlier in this session, chemotherapy drugs often have side effects, and those side effects can limit the doses that we use for patients. What you may be less familiar with is the fact that many of the chemotherapy drugs we use today are actually natural products from bacteria. So here we see just a slice of the diversity of soil organisms that are producers of medicinal products that we have today. If one of these organisms is producing a chemotherapy drug, then the other bacteria around it may be resistant to that drug so that they can continue to survive in the same environmental niche. So our goal is to exploit the natural resistance of these bacteria to chemotherapy drugs. And so I love the concept of creativity coming from ourselves, or here's creativity from microbes in terms of the different biotransformations that we can manipulate chemotherapy drugs with. I have already identified resistance mechanisms that completely inactivate 3 different chemotherapy drugs. What I want to do is target this drug inactivation to those tissues in a chemotherapy patient that do not want the drug present, those sites of the most off-topic effects. For example, the human gut. What I will do is I will take my resistance genes and clone them into probiotic bacteria. A chemotherapy patient can then consume this probiotic as a co-therapeutic to the chemotherapy that they are receiving. The molecules inside these bacteria, these enzymes, are capable of transforming the drug from the toxic to the non-toxic form. But that transformation will only occur where the bacteria are present. So in this way, when a person is treated, the chemotherapy drug will still go into the tumor and have a beneficial effect at ceasing and finishing the cancer, but we're going to eliminate the side effects to the gut area. So the person will eat the probiotic, and only there, in the site of the gut, will the drug be inactivated. So the benefit here is that the person is receiving a normal chemotherapy drug to get rid of the tumor, but we're going to reduce these gastrointestinal side effects and make sure that the gut microbiome remains undisturbed by creating bacteria that are non-pathogenic, but able to detoxify the drug to prevent this side effect. Thank you.
Could this also be used to combat side effects of antibiotic treatment?
Theoretically, a microbiome like this that was engineered to be resistant to drugs should reduce the incidence of Clostridium difficile, but I think we have to tread carefully, teaching microbes to be resistant to too many drugs, so caution.
How much time do we need to make it happen?
There was one question earlier.
Oh, I see.
How will these bacteria affect the balance of the gut bacteria in the world?
The new bacteria we will select to be those that we know from microbiome studies that are most beneficial for humans already, so it should not be disturbing.
Thank you very much.
Thank you.